All right, let's go ahead and get started. So we'll have time for a lot of discussion, lots of feedback. It's my name is Bogdan Pashunik. Welcome to our CTSI Distinguished Speaker Series today. It's my great pleasure and honor to introduce to you, although Professor Cox needs no introduction. Usually, I spend some time giving you some of the highlights, and since we're in ChatGPT space, I use ChatGPT. But as I can show you, it's like 4 or 5 pages of words and accomplishments of Dr. Cox. Nancy is a professor at Vanderbilt. She's been doing amazing science for the past couple of decades. Along science, she's been. I'm not going to mention how many decades, Nancy. Along science, she's been also serving as a visionary in genetics. She's been training a lot of, and she's been promoting a lot of the new generations of genomics or genetic scientists. So, all the way from, the training time at Chicago, as well as Vanderbilt, as well as through her leadership roles in consortia that have really shaped a lot of our understanding of how genetics impacts human phenotypes in general, anywhere from GTEx all the way from nowadays to the polygenic risk score consortia. Nancy's been more recently has been leading the Vanderbilt Genetics Institute, or she'll tell us more about it, but she's leading the charge into pushing genetics towards clinical interpretation or clinical utility in this area that we're all super excited about with precision medicine. So without further ado, please welcome please join me in welcoming Dr. Cox.
Thank you so much. And it's really a pleasure to be here. So I am going to talk about, let's see. Yeah, yes. So I really want feedback on the things I'm going to talk about today, because this is really a different space for thinking about genomics coming into medicine. And the overview is in part premised on the observation that as genetics comes into medicine, all of you are going to be, every place is going to be the kind of biobanks we already have. So you guys have a biobank here at UCLA, we have one at Vanderbilt, but all medical centers will in fact be biobanks, and it'll be possible to learn much more when we have the genetics with the electronic health records in more people. And yet we all appreciate that genetics coming into medicine is creating new kinds of health disparities. So there are health disparities that arise from common variants in the space of polygenic risk scores. It's the reason for the existence of the primed consortium, which is all about trying to develop better quality polygenic risk scores for diverse populations. But it's in the N of 1 space as well, with how many more VUSs we have in diverse populations than in European populations. And so the downstream consequences to genetic interrogation of our unequal health systems are creating new kinds of, mean that with genetics, we're creating new kinds of health disparities. But what I want to emphasize is that there's a lot of things that we can do now in medicine to reduce institutionalized health disparities that we have lived with for a long time and just didn't appreciate until we had more information on the genetics, how this was all working. And that's what I want to draw attention to today. And the ways that we would think about fixing that, which we have to do, we really have to fix this stuff, it's complicated. And what will work best for physicians is something that I think we all need to think about in a pretty nuanced way to know how to do the right things to really reduce the issues, the existing institutionalized health disparities that we have inadvertently created over time. So our biobank is called BioView, and for biobank at Vanderbilt University, we've got now about 300,000 samples. Our phenome all comes from electronic health records. So unlike the UK Biobank, where they actually ran a bunch of cheap and easy to run biomarkers on everybody and collected a lot of inventory data, and then also have access at least to the inpatient EHR data, all of our phenome is actually EHR driven. Vanderbilt built their own electronic health records more than 30 years ago. We went to Epic in 2017, but on average, we have 10 to 15 years of electronic health records data on all of the subjects that get their medical care at Vanderbilt. And so that's true for the 300,000 that we have the DNA samples on as well. And we have genome interrogation now on about half of that sample, a little under half, but all of it is being sequenced now through the usual pharma consortium. And so we'll have DNA sequence data on 250,000 to 300,000 people over the next couple of years. We do a lot of imputing of certainly transcriptomics and now more and more metabolomes and proteomes into the biobank so that people can understand how the genetic component, genetically predicted transcriptomes, metabolomes, and proteomes impact on the entire medical phenome. And this is a nice way to think about trying to interrogate the function of genes for the medical phenome, because you look at how the natural variation that comes in through the genetics relates to these biomarkers. And of course, the 3,000 that we have, the 300,000 on which we have the genome data is only part. There's another 2.8 million subjects that have a medical home at Vanderbilt, where we have just the EHR data. And honestly, so I went to Vanderbilt because of the biobank, because it was the first time I thought I could do my science better someplace than the University of Chicago, where I was for 28 years before I went to Vanderbilt. And it was, and the biobank is better than I ever imagined in terms of the richness of the science you can do. But the bonus in that has been the value of these 2.8 million with just the EHR data, because that is an amazing additional resource that I hadn't appreciated would be so valuable for the genetics end of things. And I think as we learn more about pleiotropy and the relationships of medical phenome to each other, different aspects of the medical phenome to each other, this part is something that we really need to use more of. And we've now gone in both directions, made discoveries in just the large scale phenomics data that we can then extend and understand better with the genetics information, but also vice versa, starting with observations made at the level of genetics that we then can extend in really interesting ways into the 2.8 million in which we don't have the genetics data. And I think this is a place where people have felt that value also. There's been a lot of work here for quite some time in just the electronic health record space. And I think that makes this a parallel universe for you guys. There's so much value in that, the richness of that data. And so, as I say, we already appreciate that introducing genomics into medicine has increased health inequities in predictable ways that have to do with the unequalness of access to healthcare in our systems, and the fact that so wealthy people are going to be able to get genomes in any case. But it's also a legacy of the investments that NIH made decades ago in collecting genetic information on populations. And so in the PRIMED consortium, we work with polygenic risk scores. And so the polygenic risk scores that are built so primarily with European heavy ancestry data, are less well predictive of disease in other populations. And that could hardly be a surprise. But there are other reasons that polygenic risk scores are complicated and interesting and difficult things to think about for coming into medicine. When we think of the whole distribution, what we're actually doing with polygenic risk scores involve tail properties. I mean, we're really thinking about the people who are at much higher risk for disease, often comparable to the risk that people with monogenic disease have for bad outcomes. People in these far tails may have comparable levels of risk, but of course, tail properties of distributions have their challenges. We're less confident about the accuracy of predictions in these far tails of the distribution. And of course, those tail properties are compounded when we move away from our reference distributions, as we do when we project into populations that are poorly represented in the samples in which we build the predictive models. Moreover, the United States is a really interesting melting pot. And today we are barely, so about 40 to 45% of Americans have mixed continental ancestries. And that is the part of our population that is growing most rapidly. And I mean, there is nobody my age that I know of who doesn't have or will not have, won't have continentally admixed grandchildren. I mean, I will. I have continentally admixed great niece, grand nieces and nephews, and I will have continentally admixed grandchildren. And that's the rule, not the exception. And because so little of our initial data included admixed individuals, continentally admixed individuals, our methodologies for doing these things well in people with mixed continental ancestries are not where they should be. It's the lack of data to stimulate the methods stunted the development of the methods. When we didn't need to have methods that respected the complexities of continentally admixed individuals, we didn't develop those methods. And in the United States, that's a really bad thing. Over time, the majority of our population will have continentally admixed genomes. And so we ignore that at our peril if we think we're going to use this in medicine. In the rare variant space, it's all over, diagnosing infants in neonatology units is now common to have genetic testing. But it is definitely true that people of diverse backgrounds do not benefit as much from these investigations as people of European ancestry. So certainly people who come into our breast cancer clinics, for example, who are white Europeans ancestry, walk out 80% of the time with only unambiguous results. They have only benign variants, or they have a pathogenic variant, and they have no uncertainty that they have to follow up with. But that's true for a much smaller minority of our Asian and African-American patients, Hispanic patients walking out of those clinics. They have many more variants of uncertain significance and the majority of time do not have only unambiguous answers. So we have these health disparities now that genetics are coming into medicine that we know and understand and should have expected from the way it worked. But what I want to draw attention to are a different kind of health disparity that is basically created by physician expectations about what laboratory values are. We use lab tests every day in medicine, and they're a real backbone of diagnosing and treating disease. And so we've spent a lot of time on lab values. So one of my senior postdocs worked with Leah Davis, a faculty member at Vanderbilt, and one of her graduate students to really develop a pipeline for really cleaning the laboratory values. For those of you who don't know, the units in which the lab values are reported in change in an arbitrary way as the lab technologies change. So the units that are reported in change. When you move to a different lab, the whole thing may shift a bit, and over time we may unshift that. But so cleaning the laboratory values was a process, but they got to a point where they were pretty confident because they could show that the same GWAS top signals were obtained for our lab value data that are obtained from research quality data for the same values, the same laboratory measurements, and the same kinds of heritability estimates were obtained and so forth. And so that pipeline is publicly available for cleaning, which has some utility for biobanks. But it allowed us to look at laboratory values in a much more comprehensive way, which we've been doing now for some years. So how many people here are physicians? Probably a minority. Yeah. So when I do this for a physician-based group, and I did it for our grand rounds recently at Vanderbilt, because I wanted physicians to be thinking about this. What proportion of the lab values that physicians order and interpret on at least a monthly basis are heritable? And so when I ask, I start at 10%. How many think there are at least 10% of the laboratory values have what we would say is significant, statistically significant heritability? And actually, only about half the physicians think that even 10% would have heritability of, that at least 10% of lab values would be heritable. And if you go up to 20%, a lot of the hands drop out, and nobody thinks that more than 30% of lab values are heritable. But all lab values are heritable. I mean, your geneticists, all of them are heritable. And this is a sample size issue. And of course, yes, the heritability can get very small when the sample sizes get very big and still be statistically significant. But the bottom line is 30 to 40% of labs have what anybody would call quite substantial heritabilities. So 50% or higher, which is shocking to physicians. And because physicians think of laboratory values as dynamic measures of disease risk and progression, and that's how they are used. Most of the lab values that get ordered on a regular basis are capturing bad stuff. The disease is happening. We're following the progression of disease, or we're worried that cytotoxicity from chemotherapy is leading to low white cell counts and will cause an infection. And so we're following those white cell counts to make sure that people are not in danger of developing infections while they're taking certain drugs. It's the dynamic part of the disease process that we're really looking at with these laboratory measurements. And they are dynamic. They are changing as disease happens. So the problem is that when there's such a strong genetic component to so many of them, and a reasonable genetic component to most of them, that part is a drag on the dynamic parts of the measurements. And while the G by E may be somewhat more dynamic because the measurements are changing, it's mostly the E. It's mostly the internal and the dyshomeostasis of disease that is leading to those changes. And as we're going to talk about, the bottom line is that the genetics may lead to a lot of misstaging of disease, so that we know it's changing. We know we're in a process where it's changing, but we mistake the starting value, the first one we see that seems abnormal, for absolute truth on some arbitrary spectrum of reference ranges that we need to be thinking about. And so once you know how heritable lab values are, well, that leads to the obvious question of what proportion of heritable labs have significant differences in means and or variances by EHR reported race or ethnicity. And, you know, it's about half of them. And again, like, this is just the means, especially for populations of African ancestries. We often, and there are about as many laboratory values with mean differences where African Americans are still more likely to be outside reference ranges at both ends of the distribution because of increased variance than are outside at only one end of the more outside on only one end of the reference ranges. So the variance issue is not a trivial one, and especially in populations of recent African ancestry that have much more genetic variation, much more functional genetic variation that impact laboratory values. And as we'll discuss, you can't get away from the fact that blood, because it is a convenient tissue in which to make measurements, but is also a major place where pathogens enter the body and do their worst. All of our blood-based measures are inevitably affected by adaptations to high pathogen environments. So we end up having many more large effect and common variants that really can increase heritability. Part of the reason so many of these lab values look pretty heritable is almost certainly due to the fact that pathogens invade through blood, and we try to adapt to those things, which creates this extra layer of heritability that is not small. Variants with large effect that are common drive a lot of heritability. And we'll come back to that as well. So I want to start in with the examples now. And Duffy is the first primary example because Duffy is a truly benign variant that affects white cell and neutrophil counts in a big way. So has a huge effect actually on white cell and neutrophil counts. And so, and work, Jonathan Mosley conceived and has led now for some time. He's been looking at the consequences of Duffy in EHRs. And then the first thing they reported is, yes, you would think that all physicians know about benign ethnic neutropenia and do not order bone marrow biopsies when the only indication is low white cell count. But you would be wrong if that's what you thought, because they do. And when they do, in people who are Duffy homozygotes, there is no return ever on that bone marrow biopsy. There is no value because it is always negative. It is always negative. And so that's what this first paper showed, and it was reproduced in other healthcare centers. When the only indication for the bone marrow biopsy is low neutrophil, low white cell counts, in people who are Duffy homozygotes with genetically low white cell counts, the results are never positive. And a bone marrow biopsy is not a trivial thing. But of course, the problems with Duffy are not just bone marrow biopsies. So Cecilia Chung immediately said, oh my gosh, we look at white cell counts when we treat patients with lupus, first-line meds, azathioprine, and we take people off those meds when white cell counts are low. And so, yes, African-American women who are Duffy homozygotes in this study got something like 20 to 30% of first-line meds relative to other African-American women who are not Duffy homozygotes. And when it was a hematopoietic toxicity that was the cause, so the Duffy polymorphism was responsible for all of that when it was the hematopoietic toxicity. When it was something else, there's no effect of Duffy. So again, if it's just hematopoietic toxicity, people are being yanked off of first-line meds, for lupus and other autoimmune diseases when they don't need to be. We've always known that African Americans with lupus, and it's one of the few autoimmune conditions that they're at higher risk for than Europeans are, they do worse. Some of that is APOL1 and the kidney consequences of APOL1, but some of it is undoubtedly not being able to take the first line medications. And so they're doing that now. They're doing those hard studies where they try to understand how much of the adverse outcomes relate to not getting as much of these first line therapies. And that's much harder research to do, but they're doing it. But it's like, then you think, well, what about cancer chemotherapy? What about the therapies we give to transplant patients? And yes, it's a problem in all of those things. And it's a little bit shocking. So there's a ton of repeat testing that minority populations bear the vast burden of. Unnecessary bone marrow biopsies, under treatment for lupus. But part of the reason African Americans didn't get into so many of the cancer chemotherapy trials is that they have a threshold on white cell counts that African Americans who are homozygotes will rarely meet when they're healthy, let alone after they're being treated for cancer. And it's benign. The white cells are hanging out in peripheral tissues. They're there. They don't have a higher risk of infection with these low cell counts. The white cells are there. They are just not in circulation. That's why this is a benign polymorphism. It is not that it increases risk of infections in any way, shape, or form. So there's a lot of people, African Americans, homozygous for Duffy, being taken off meds in all kinds of contexts where they don't need to be. And this is crazy. This is an institutionalized health disparity that we know how to fix. It's a big effect, common variant. But I will tell you, the genetics of white cell counts in anybody, in white people, is not predictive of infection, but it is predictive of being taken off meds because people are worried about infection. There's a lot of heritability to these cell counts that is not relevant to why we use these laboratory values. So yeah, transplantation, chemotherapy. For many years, to do an appendicitis, people had to reach a certain white cell count to show that they were in danger of bursting their appendix. And African Americans, not surprisingly, at that time had then more burst appendix because Duffy homozygotes just never got to those. They got to really elevated white cell counts for them, but not to the threshold that was required for surgery and had more burst appendicitis. So it's everywhere embedded in our healthcare system in crazy ways. And race-based reference ranges cannot solve this problem. It is not about whatever it is that people say when they mean when they say race. And it's certainly not about skin color. It depends on what alleles you have at this polymorphism and at some others that have big effects on white cell counts that don't predict infections. So certainly there are large effect common variants in African Americans that have a big effect on white cell counts. This is like a monogenic, a Mendelian type thing for white cell counts in African Americans because it accounts for 10% all by itself, basically, of the heritability in African Americans. But there's plenty of other genetic variation that is affecting all of us all the time in the way these get interpreted that is not helpful. The genetics of white cell count is not predictive of the reasons physicians are looking at white cell counts for medicine. But it's not just stuffy. So here's G6PD. So G6PD affects hemoglobin A1C levels, has been known to affect hemoglobin A1C levels for some time. But I don't think until recently people have been in a position to look at the full consequences of the ways that G6PD affects hemoglobin A1C levels. So why does G6PD just being a carrier of G6PD variant, well, it's on the X. So in males are homozygous. So the problem is that there's red cell hemolysis. So the red cells turn over faster in people with G6PD variants. And hemoglobin A1C is a measure of how much of the hemoglobin is glycosylated. So you're really looking at how much sugar it ends up being glycosylated, because that's a measure of the sort of average circulating level of glucose in the blood over about 3 months in people without G6PD variants, but over maybe 1.5 to 2 months in people with G6PD variants. So that's why the values are nearly a full point lower in people with diabetes who have G6PD variants versus African Americans who have diabetes and don't have G6PD variants. And here's the problem that makes these guys look like they have much better control of their blood glucose levels than they do. Because the reason their hemoglobins have less glucose on them is not because they had lower levels of circulating glucose, but because you're looking at an average over a shorter period of time for that glycosylation to have occurred. So we've known this for a long time, but we don't always have information on diabetic complications in the cohorts in which we had measures of hemoglobin A1C for genetic purposes. And so in a biobank, we can say that overall there's a 1.2-fold increased risk of any complication to African Americans who have G6PD variants relative to African Americans who don't. But it's especially serious for diabetic retinopathy, which circulating blood glucose was known to be the major risk factor for. Increased risk for ketoacidosis, even in type 2 diabetics, because they've got much higher circulating levels of glucose than is appreciated. And if they get the flu, they're much more likely then to develop ketoacidosis. In fact, it's an increased risk for all diabetic complications other than peripheral vascular disease, which we already knew was not a major consequence of high circulating sugars. And this is just in BioView. I will say also, the more covariates we put in that appreciate the length of record for how long somebody's had diabetes, BMI and other confounders that are predictive of the development of complications, the worse this gets. So higher levels, higher odds ratios, the more of those covariates that you put in. So now we've got a consortium, a worldwide consortium, looking at this in biobanking data. It's a project that's been approved and primed so that any of the biobanks can join in, or we'll be trying to look at this at scale. But and so what's happening here is that both physicians and the African American patients who have G6PD variants are being fooled by how good their glucose control is. They are possibly not being diagnosed as early. Our preliminary data suggests that that is the case. They think they're doing better than they are, and so they're not being treated as aggressively as they should for their actual levels of circulating glucose. And hemoglobin A1C, I mean, doctors get penalized if they treat diabetics and don't get this hemoglobin A1C value and use it to modify treatments. This is the way diabetics have of getting the best control of their glucose to reduce the risk of complications. And for these people, it's misleading in a way that we can absolutely predict and correct for. There are other ways that are much more expensive of measuring glycosylated hemoglobin that gives you accurate results even in people with G6PD. But they are much more expensive. And so arguably trying to fix this so that it doesn't continue to do this kind of damage is something that we need to be looking at. And as I said, alleles with large effects that are not rare, and these are not rare. G6PD, so that was just one, and that was only one of the G6PD alleles. So we just grabbed one for demonstration purposes. But there are several that are collectively quite common across world populations with exposures to malaria. And in particular, there are a lot of Asian populations which have collectively high frequency of G6PD variants. Virtually all of them have this consequence. And so again, so this is something that is highly relevant to a lot of the non-European populations that we treat every day in medicine that we should be doing better with. But as geneticists have sometimes been known to say, it has not escaped our attention. Again, this is an adaptation to a high pathogen environment. And we probably need to be a lot more thoughtful about all of the alleles that have been characterized to reduce the risk of malaria and other pathogens, and look at their consequences for all the measurements that we make in blood. So we're doing that in a systematic way with ApoL1, but also several, the sickling allele actually also has some effects on hemoglobin A1C that we probably need to look at in conjunction with G6PD. Again, a variant with a not small effect that is not rare in our African-American population and is certainly not rare in Africa. So what do we even call this kind of disparity? So that with Duffy, it's particularly galling. This is a genuinely benign polymorphism. It has no ill effects on health that we know of, but it certainly has ill effects on African Americans in our health system, and frankly, possibly on Africans in Africa, because they use the same reference ranges worldwide that were built decades ago in what we would now consider to be ridiculously small samples of white men. That was shocking to me. And I'm sure they make allowances all the time in Africa for the variation that they know and understand. But honestly, and so I'll bet you the rate of bone marrow biopsies in Duffy homozygotes is less, but I'm not sure that treatments for autoimmune disease, cancer, transplants are always being looked at in the same way with respect to white cell counts there. And so we've got some collaborations in Africa to really try to look at this in more detail. It's like, it's our ignorance of the consequences of these variants and their effects on laboratory values that are at the root of this. So it is genetic variation, but it's often totally benign to why we're making the laboratory measurement, unpredictive of any of the outcomes, but it affects the measurement itself in some way that then is misleading to physicians. And they certainly become institutionalized health disparities, but they're not really genetic causes of disease. They are genetic factors that have a big impact on laboratory values, but are not impacting the reason that we make the measurement. And that's why the last example I'm going to give is on kidney function measures, because this is one of the best examples I've ever encountered of geneticists and nephrologists talking past each other, because I don't think I don't think when geneticists use genetically predicted estimated glomerular filtration rate as a measure of kidney function, that they have any idea that cystatin and creatinine, which are so cystatin's a protein, creatinine is a metabolite. I don't think they have any idea that that protein and that metabolite don't have anything intrinsic to do with kidney function. Nothing. They just happen to be recycled through kidneys, so that when filtration is failing, their values change. But we are looking at basically a steady state serum level of these values, and assuming that the only thing that affects those values is kidney function, sex, and age, because in the equation, those are the only variables that go in. And yes, for creatinine, there used to be race because there was a big average difference in creatinine levels. And guess what? There's also a big difference in the variances. But it's not about race, it's about what alleles you have, because cystatin C is highly heritable. Creatinine is quite reasonably heritable, but they have nothing intrinsic to do with kidney function. They're just cheap and easily measured. One's a protein, one's a metabolite that get recycled through kidneys. Their values change when kidneys begin to fail. And we use these measurements as if, these serum measurements, as if the only thing affecting their levels, their steady state levels, was kidney function and sex and age, which are the only other covariates that go into the equation. As if. So here's the cystatin C raw protein, value that gets measured, and the cystatin C estimating equation for EGFR in the UK Biobank data. And we had to take out a 1 megabase region on chromosome 20 that has the cystatin C structural locus, which has been known since the early days of Framingham to account for 80% of the total 60% heritable trait that cystatin C is. So that structural locus has a huge, it just makes it messes up everything that in the rest of the genetics, because it's got such a high p-value association. But there are plenty of biomarkers, plenty of genetics to cystatin C. So this is the same thing for creatinine, but with and without the race correction, which of course this was UK Biobank, so it doesn't even matter. But it's a one-to, because the equation is just using the measured level, of course, the genetics for EGFR and creatinine itself are the same. And the physicians will say, well, yeah, okay, so they're heritable. We only care about what's happening when these values are declining, because then we know people are in trouble. But declining from what? Because there are plenty of people on transplant lists who've been on transplant lists for 5 years in stage 4 kidney disease that haven't progressed. And there are people who die of kidney failure before they can get on transplant lists from stage 1 disease. And I can tell you from the in silico work we've done in BioView with a different way of measuring kidney function that regresses out the genetics of creatinine and cystatin C that are not shared and are the only things that tell you about kidney function. That's misstaging. That's genetics misstaging where people are in the disease process. And the very high heritability of cystatin C makes that a not uncommon thing. But creatinine is the more commonly used biomarker for this. But it is possible, and part of it is the fact that it's often the case that the first physicians don't order these kidney function tests until they have a suspicion of kidney disease. And so if you have hypertension or diabetes, they'll start ordering these kidney function tests on a regular basis. And the problem is that there will be some people in stage 3 or 4 kidney disease just based on their genetics that will appear to be in stage 3 or 4 kidney disease. There's enough genetics that even though their adjusted value would be mid-range, the genetics is enough to place them into those ranges. So the problem is in the staging and the way it affects the staging. So this business that creatinine and cystatin, the only parts of their biology that are about kidney, they have nothing to do with each other. They both are just among the many things that totally get filtered through kidneys. But there's a lot of, so there's some shared genetic architecture that's very obvious, but there's a lot of unique genetic architecture that is not shared. And all of that is noise. All of the genetics that is not shared between those biomarkers is noise for how we think about the genetics of kidney function. And there's a lot of noise here. So the genetics of creatinine is so not just about kidney function. It's about a lot of other things. That is interesting biology, but not telling you anything about kidney function. And so you could imagine a completely different way of thinking about this, where we use the shared genetic architecture between the two to really get a better understanding of at least filtration-based kidney function and who's at risk for it from the beginning. But you could also imagine a different kind of biomarker that regresses out the genetics that is not relevant to kidney function from either of these measures, and then you'd have a much more accurate measure of kidney function that would be much more relevant for the staging of disease. And I think, like, this is the sort of thing when we fully twig to all of our, as geneticists, misunderstandings of how this worked, and all of the nephrologists. So we're working with a team that, that's made actual measured really measured glomerular filtration, and then worked on developing the estimating equation. So we're trying to use all of that data now. It's like, how did we misunderstand each other for so long and then not fully appreciate how genetic these things were and how that genetics was going to play out in the staging of disease, and us thinking that EGFR, that a polygenic risk score for creatinine, EGFR, was going to tell us all that much about kidney function. It's just like, geez, we could do so much better. So yeah, there are no simple solutions to complex problems. And these are really iceberg problems. So the things I've been talking about, like, this is the very, very tip of a big iceberg. And we know what happens. We ignore icebergs at our peril, right? We know what happens. And all of our thinking about the solution has been driven by some of the work that we've done. And you guys here think about all the time too, that relate to the way we think about biomarkers like transcriptomics, and how we think about, okay, you've got this genetically regulated part of, so we're interested in diseases and traits. We can measure something like the transcriptome. We hone in sometimes on just the genetically regulated part because that's got a one-way arrow into our trait. And we know that what we measure is influenced by reactions to having disease. So the feedback, when we look at all of the different genes that are differentially expressed in kids with asthma and kids without, or worse, in people with type 2 diabetes and without, in adults that have been out of normal glucose homeostasis for decades, there are a ton of genes that are differentially expressed that are all a consequence of having this disease and the body trying to fight back to healthy homeostasis. And for genetics, we're much more interested in the causal space, but there's a lot of value in many ways in other contexts. So certainly in cancer, we often, when we look at cancer transcriptomics, we use the genetic information from the person to regress out the expected transcriptomics to highlight all of these things that are being influenced by the mutations and other things. And when we think about moving forward to the multi-omics space, where we hope to use epigenomics, proteomics, metabolomics, and the transcriptomics to understand more of the environmental exposures and social determinants of health and how the consequences of those exposures affect all of these biomarkers so that we understand these better. Yeah, we will often want to regress out the genetics to see these parts better, to see the consequences of exposures better. And so I think that's what we have to think about for laboratory values if we think about solutions. So the problem is it better to remove the genetics from the biomarker itself, the genetics that are irrelevant to why we use it? Or do we think about individual reference ranges? And this is one thing that I think when I talk with physicians about it, there are some thoughtful physicians that I respect who say physicians will learn more over time if we do this as individualized reference ranges, because they will start to see how variable certain of these are, how much genetics is influencing how we should be thinking about reference ranges. There's no better way to teach physicians, in some sense, population genetics, than letting them see that as a part of their regular care. And then there are other physicians that I highly respect and love, like my husband, who say, are you nuts? You think I don't have enough to do? I have these reference ranges in my head. I can't individualize reference ranges. No. And the truth is, as I said to some students, I wish that we could move away from these are bad things, these laboratory values that we use, because it means we've already lost. People already have kidney failure. And I hope for a world in the not too distant futures where we're making multi-omics measurements every year on people and trying to catch them before they slide completely out of healthy homeostasis and just pull them back in. But I'm obsessed with getting this fixed before I retire, and I'm old, because we can't tolerate the degree of institutionalized health disparity that we already know about. And the depth of it is way worse than I can tell you now from this. Just the number of people on transplant lists that will never progress break my heart, for the number who are going to die before they can ever get on a transplant list. Like, that is creepy. And we shouldn't as a society have to have those kinds of inequities if we can fix it. So what is the right solution to this? And how do we do better for our patients with the understanding of genetics we have? So I hope there'll be questions. So a lot of this is very strongly influenced by, so when I first got to Vanderbilt, the first person that I never met before was Consuelo Wilkins, who wanted me to work with her on some program projects in health disparities. And so we've had a collaboration ever since. And Roy Weiss was my colleague at the University of Chicago, who became chair of medicine at Miami. So we have Hispanics from Miami. We've always had a long-term relationship with Meharry. And so we have a really diverse population that we work with in these, and now in our second round of 5-year funding for the Southeast Collaborative for Innovative and Equitable Solutions to Chronic Disease Disparities. But also a lot of this was strongly motivated by our Primed grant and about all of the work that we do in Primed, where a lot of the quantitative measures that we look at are the laboratory values, and the opportunities for doing good within Primed in this space, I think are really there. And so it's another it's another way to think about using genetics to improve health inequities instead of just exacerbate them, which some of the other ways of thinking about polygenic risk scores actually do exacerbate them. This is one way we can try to correct some of the health disparities that already exist and our funding bodies. So happy to take questions and hope you all have some.
We'll have time for some questions. There's some comments on the chat.
Oh, good. Okay.
I don't think there are questions that will.
Okay.
Okay, Dan?
Yeah, and I don't mean to, like,
Maybe you want to wait for the microphone?
oh, I have to wait, sorry.
I can hear him, but the people online won't be able to hear you otherwise.
Yeah, I mean, this is absolutely, I mean, I think it's actually what you were showing with the kidney work and kind of using the cystatin and, the individual stuff. That's absolutely brilliant. We have to begin to think that way. It's a great idea. It's spectacular. I have kind of like a much less brilliant kind of thing, that I was thinking about as you were talking, which is that, there's so many lab values that we know are different between males and females, for example, like potassium, even like simple things like potassium, stuff like that. And I just wonder, and so there are two parts to my question. So you talk, and obviously you've taken a genetic approach to this, which is appropriate. So, shouldn't we normalize the laboratory values in our health records for males and females, and and kind of report them that way? I just don't understand that, and hopefully down the road we'll be able to use the genetic data from individuals to actually normalize their lab values based on what you're kind of showing. So that
honestly, I think there's no better way to get genetics into medicine, really, to get it into medicine than to fix this, because it's not the repeat testing alone, the cost of the repeat testing alone would more than pay for what you need to the genetic interrogation that you need in order to correct anything at the lab value level.
Well, the other thing that we didn't really talk about today was trajectories. So
Yeah.
for example, again, my silly example of potassium. I just remember like being an attending and going around and, in the hospital, people's potassium, it's like once the potassium is low, somebody is like total body depleted and you're like catching up and it's this issue. And it's just like such a simple thing. Everybody uses like the lab value to show that it's low, but like, shouldn't we have predictive algorithms to tell us?
Yeah. No,
It's so easy to tell us.
And when it really matters. Yeah, because that's the thing. So a polygenic risk. So yeah, and a good example of, so this is not just about genetics. Sex is an issue too. With some of the first lab values, one of the grad students was fooling around with polygenic risk scores, and a polygenic risk score for alcohol use disorder, for alcoholism, was predictive of liver transplants in males, but not females. Why is that? Because liver function tests have big sex differences in their values that were never corrected for considering who would get on transplant lists. So we worked with the transplant scientists at Vanderbilt to devise an algorithm that would take that into the sex differences in those values more appropriately into account in allocating liver transplants, that suggested through simulations that the death rates for people not getting transplants would go down in females relative to what they are now. But it's a crazy thing to just think that, that sort of polygenic risk score would be predictive in males and not females. And it wasn't anything to do with alcohol use per se. It was the liver function stuff that was the heart of that, which you could show that analytically. So go ahead.
So beautiful work and horrifying. My question is, when it comes to solutions, the things that you mentioned and the things that I can think of, having a baseline measure for people, having repeated tests, having genetic information for patients. All of those are things that either insurance doesn't cover or that people without insurance definitely cannot afford. So if we're trying to reduce health disparities, a lot of the people that we're trying to reduce the disparities for will not have access to those solutions anyway. Do you have?
Yeah, so I have some ways of getting this in front of insurance companies. So Jackson Labs runs a meeting every few years with a bunch of the insurance company executives and with geneticists. And I've already twisted some arms, so I will be on that agenda the next time there's a meeting. And the insurance companies are all about what it is going to cost them. The savings from this are substantial, and this is. So we are also working with our health policy and health economics people, because the savings from this are consequential. The cost savings to the reduced repeat testing, but also reduced numbers of bone marrow biopsies, a potentially better outcomes for people with autoimmune disease and cancer and transplantation and diabetes. Reducing diabetic complications alone, to the degree that we already can see these G6PD variants have effects, would pay for anything. I mean, would pay for, we could do whole genome sequences and build build all the corrections from that. The cost of the sequencing itself or genotyping a cheap array is nothing in comparison to the costs from just the repeat testing is crazy, and a lot of that falls on the hospitals themselves, frankly. And then also the worst consequences of these things are not cheap, are very bad. So it really would improve health overall and increase equity. And I think even insurance officials would agree. So I harbor the delusion that they are convincible on economic grounds. We just need to provide them with the data, and we're working really hard to get that data together, because I think without the without sort of economic proof of value for the proposition, yeah, it would be a heavy lift, but this is bad stuff, expensive, bad stuff.
Great. On that note, let's thank Nancy again.